I'm Müller, and today I want to present my master thesis, a comparison of underwater 360-degree video monitoring and diver-based visual census using reef check methods in the Red Sea in Egypt. This study was a pilot study to investigate the usability and feasibility of 360-degree video underwater monitoring for fish, invertebrates, and substrate. 360 video technology is emerging, and the cameras are now comparably cheap and accessible for private people, but there is almost no research on the use of 360 video for underwater monitoring. So with this first pilot study, I had the aim to take first steps in using 360 video for underwater reef monitoring. I wanted to find what are the advantages or disadvantages compared to diver-based visual census. And I expected that many more questions would open through this pilot study because it is an almost not investigated field yet. So I wanted to define recommendations on which aspects of 360 video monitoring there should be further research on. I think we all know that it is very important to choose the right method for your data collection, since it can have a strong impact on your data. But you need to know the method well when you're choosing. So 360 video monitoring needs to be investigated and understood in order to be used. And last but not least, I also wanted to investigate the time efficiency of 360 video monitoring compared to the diver-based visual census. I was working with these two partners, the Red Sea Environmental Center from Dahab, Egypt, who did the diver-based visual census data collection for my study, and with Sign Planet, who specialized in immersive media creation for marine conservation and who collected the 360 video footage. For the data collection, we followed the Reef Check Protocol, meaning we deployed a transect line in the reef in either 5 or 10 meters depth. This transect line consisted of 4 segments that had a length of each 20 meters, and between each segment there were 5 meters that were not surveyed. And we surveyed 3 different categories: fish, invertebrates, and substrate. Not all organisms were counted that were filmed, but only indicator taxa from the Reef Check Protocol. So for the fish, they count 6 classes and additionally 3 species, and for invertebrates, they count 3 classes and 7 species, and the substrate is categorized in different categories. And the divers noted their observations in the field then on waterproof field slates. The substrate was just measured at every half-meter mark of the transect line. So the divers just went along with a plumb line for the DVC, and the camera operator went along and basically dipped the camera close to the transect line at every half-a-meter mark. For the fish and invertebrate survey, we monitored a belt transect of 5-meter width. On the pictures, you can see how the DVC was performed, always with 2 divers per survey. And the upper one shows the movement for the fish survey, where 2 divers swim next to each other in zigzag lines over the belt transect. And at every 5-meter mark and at the beginning of each segment, they hover for 1 minute and count the fish in the water column. And then they move forward to the next 5-meter mark. For the invertebrate survey, the divers split. So each monitors only 1/2 of the belt transect, and they swim in synchronized zigzag without stopping. When we collected the 360 video footage, we combined these 2 methods in one recording, and the camera operator just swam straight along the transect line and zigzagged only the camera over the belt transect. So we collected per transect 2 360 video files, one for fish and invertebrates, and the other one on the way back for the substrate. For the 360 video monitoring, we used the Insta360 One X, and the 360 video analysis was performed in Insta360 Studio. The analysis of the data that was then compared with the DVC collected data was all conducted by me. But to make sure that observers' bias did not affect the results, a reef check diver from the RSEC also analyzed 2 files of each survey type, but there were no significant differences between her results and mine, so I assumed that there was no or very little observer bias in the video analysis. We also measured the time needed for the data collection and the time for the data analysis. And it was measured in people minutes. So for example, if 2 people had to do a task that takes 10 minutes, then it took 20 people minutes. These were the locations around Dahab in the Red Sea in Egypt where we collected the data. In total, we monitored 9 transects in depths of either 10 or 5 meters. And we also did 5 test dives first with a camera to find best practice in handling and everything before we started with monitoring. So the results for the fish surveys were significantly different between methods, as you can see. Usually about half of the amount of individuals was detected with 360 video monitoring, compared to the diver-based visual census. However, I found that the difference of abundance and species richness was higher between survey durations than between methods. As I said, the invertebrate and the fish survey were combined in one recording, so the hovering that the DVC divers of the fish survey did to count fish in the water column was not performed in the video survey, because with 360 video you are monitoring the water column the whole time. But seeing these results, the survey duration should be standardized between methods and actually also within methods, which was not the case for both monitoring methods. However, when assuming that roughly only half of the fish was counted in 360 video monitoring compared to diverse visual census because of the much shorter survey duration, there is still one curiosity, and that is the Helium lunolatus, the broomtail wrasse, that was counted only 2 times with 360 video monitoring and 37 times by diverse visual census. And I found that this might be due to the special detectability features that this species has. With detectability features, I mean features that help you distinguish a species from another one. Sometimes it's the color, sometimes the shape, the size, a special movement. And in this case, the broomtail wrasse has a yellow spot behind the operculum, which is very easy to spot for divers. But when filming, the colors are affected by the amount of light and the quality of the camera sensor, and that can alter some colors. So it's possible that this detectability feature of this species was not very well suited compared to the other ones of the other species, because the yellow spot was not really visible in the video. So therefore, detectability features of fish in 360 video should be better investigated. And finally, there might have also been an effect of divers' presence on the fish. The diver-based visual census surveys always took place before the 360 video monitoring. Logistically, it was unfortunately not possible to randomize the order. And we kept the 10 minutes of diver-free time at the transect before we started recording. However, the Reef Check survey with the diver-based visual census went through as a group of 4 to 6 people, and spending a long time at the transect and looking very closely at the reef. That probably also affected the fish assemblage as well. Concerning the invertebrate survey, the difference was highly significant, not only in abundance, but also in species richness. And here you can see that there were definitely issues with the detectability features of some species, like the diadema and Heterocentrotus mammillatus, which were recorded 53 and 73 times respectively by the diver-based visual census, and not a single time by 360 video monitoring. So here it has to be questioned if 360 video monitoring is a useful monitoring method for invertebrates, or at least for these invertebrate taxa that were monitored here. The survey duration could not really make a difference here, because these organisms are either sessile or very slowly moving organisms. So the assemblage should not differ between the 2 methods over such a short time. The by far most counted species in both methods are Tridacna, which I excluded from the former graph because they would make all the other bars shrink invisibly. So it was probably detected so often in the 360 video monitoring compared to all the other taxa because it had a strong detectability feature. This blue color of the soft tissue of a majority of the individuals, and it was really shining in the video. The Tridacna were categorized in different size categories, and you can see that the numbers counted with diverse visual census decrease with increasing size, while 360 video monitoring has the highest count in the category of 10 to 20 centimeters. And the resolution of the video might have been a problem to detect, for example, the very small individuals, which would explain why only so few were counted. But it does not explain why, for example, these 30 to 40 centimeter individuals, or this one 40 to 50 centimeter large individual recorded only by DVC, were missed in 360 video monitoring. So this was most likely due to the fact that the transect was filmed roughly 1 meter above the substrate, but the reef structure is very complex and the individuals could be hidden behind or beneath some structures very easily. And that it worked well for the fish survey, but for the invertebrate survey, it was important to hover directly over the substrate and really follow the reef structure. So it has to be questioned in general if 360 video monitoring is suitable for these invertebrate taxa, since some of them also hide in crests and crevices like lobsters and shrimps. And the idea with video monitoring is that you can just follow a certain movement pattern like we did with a zigzag movement over the transect, rather than actively searching for organisms. So to find the organisms, the camera operator would need to know where to look for the species and then point the camera at them. And in that moment, it would probably already be easier if they just directly performed diver-based visual census instead of searching for the animals and then recording them with the camera. The Substrate Survey was the only survey with similar results in both methods. So here the 360 video monitoring worked as well as diver-based visual census, probably also because this was just a certain amount of data points that were recorded, and it did not matter how fast you went along the transect. And here, obviously, an advantage that you had was the reviewable data in the 360 video monitoring, so experts could also take a look, verify your substrate identification, which is not possible in the field. There you have to rely solely on the training of the divers. And the same, of course, holds true for the fish and invertebrate surveys. Now we get to the time efficiency of the two methods. Here you can see the average amount of people minutes needed for one transect. And the people minutes needed for 360 video monitoring and diver-based visual census differed significantly between methods. DVC needed much more time for the data collection. The difference in people minutes was mainly because of the team sizes, because 360 video monitoring was performed by 2 people and the diverse visual census was done in team sizes of 4 to 6 people. And additionally, as I already said, the survey durations were very different between methods, so the time in the field was just much longer for DVC. Of course, since the survey duration had such a strong influence on the results, it should be standardized. They should have the same duration in both methods, but then still you would have a smaller team and less people minutes required for the 360 video monitoring. And especially when taking into account that time in the field is usually quite expensive time in marine science, the time efficiency is definitely a very important point to consider when choosing the method. So as a conclusion, here are the advantages and disadvantages that I found when comparing the two methods. I already mentioned most of them during my discussion of the results now, so I will not get into detail now, but I'm happy to answer questions related to these advantages and disadvantages after the presentation. So, as I expected, this study actually opened more questions than it answered, and it also gave a good insight what needs more careful consideration. In general, the method should be tested in different marine environments, since this was now a reef, and it posed quite some problems for the invertebrate survey. But for example, sea cucumbers, where they are usually on sandy substrate, and they were found very easily. So it would be interesting how 360 video monitoring performs, for example, for invertebrate surveys on sandy substrate instead of reef environments. I could only speak briefly about the detectability features today, but they are also very interesting and important to further investigate in order to know if the method is feasible for the organisms you want to monitor. I couldn't talk about the effects of environmental variables due to time reasons. I also investigated them in my thesis, and they also should be studied further to know the best applications for the method. Also, different softwares and analyzing processes should be tested because, as I said, for this study, I used Insta360 Studio, which is a freeware. And the idea was that a license for 360 video softwares can be quite expensive. So it would not be feasible for organizations like, for example, IMBRSea, where I did the study now, to buy one just for the analysis of a 360 video. But there were some difficulties that slowed down or complicated the analyzing process in this specific software that I used that might have been avoided with other softwares. So a best practice of analyzing 360 videos should still be better developed. You might also have noticed that many of the advantages of the 360 monitoring that I listed before are actually also advantages of flat video monitoring, like non-extractive, permanent, reviewable data, etc. So a comparison between flat and 360 video monitoring should definitely be conducted to see if there is an added value of 360 video compared to conventional video monitoring. Yes, and despite all these questions that I now have after this pilot study concerning the method, I do believe that it is a promising new technology that can bring video monitoring on a new level, and I'm looking forward to keep working on exploring 360 video monitoring as a tool for marine conservation. Thank you.